Life is often described as a struggle against entropy. From the macroscopic scale of a marathon runner to the microscopic realm of a single bacterium, every living entity must pay a thermodynamic tax to exist. But what is the absolute minimum payment required? How low can the energy bill go before the lights of life flicker out? Today we are exploring the cutting-edge physics of living matter, journeying from the metabolic ceilings of human endurance down to the sub-sea floor zombie microbes that survive on energies so low they challenge our very definition of being alive. We will uncover the hidden costs of accuracy in DNA replication, the theoretical limits imposed by the laws of information theory, and the startling efficiency of the molecular machines that power us. In the grand casino of the universe, the second law of thermodynamics guarantees that the house always wins. Disorder or entropy inevitably increases. Left to its own devices, a complex structure like a cell should degrade into a chaotic soup of chemicals. To avoid this fate, life must constantly consume high-quality energy, like sunlight or chemical bonds, and dissipate low-quality energy, usually in the form of heat, to pay for the maintenance of its internal order. This is the thermodynamic cost of living. For decades, physicists and biologists have asked, is there a hard lower limit to this cost? Is there a quantum of energy below which life is physically impossible? The answer has profound implications not just for biology, but for the search for extraterrestrial life and the future of synthetic engineering. We are accustomed to thinking of life as a high-energy phenomenon, birds flying, hearts beating, neurons firing. But recent discoveries have revealed a shadow biosphere of organisms living in the slow lane, operating at energy levels that are nearly indistinguishable from death. Before we dive into that microscopic abyss, it is useful to establish a baseline of energy consumption at a scale we understand: the human body. A resting human adult burns approximately 100 watts of power. That is roughly the same as a bright, incandescent light bulb. This is our basal metabolic rate, or BMR, the energy required to keep our hearts pumping, lungs breathing, and ion gradients maintained across our cell membranes while we do absolutely nothing. In thermodynamic terms, this is the cost of fighting off equilibrium. If this power supply is cut, our highly ordered tissues quickly succumb to entropic decay. We die, and our bodies decompose. Remarkably, recent research into elite endurance athletes, Tour de France cyclists, Arctic trekkers, and ultramarathon runners has revealed a hard upper limit to sustained human energy expenditure. While we can sprint at high power for short bursts, the human body cannot sustain a metabolic rate higher than approximately 2.5 times its BMR for long periods, such as weeks or months. This metabolic ceiling appears to be determined by the digestive system's ability to process calories. No matter how much an athlete eats, their body cannot turn food into chemical energy fast enough to exceed this limit indefinitely. It serves as a stark reminder that biological systems are constrained by physical bottlenecks, rates of diffusion, enzyme kinetics, and heat dissipation. But while the upper limit is fascinating, the lower limit is where the physics gets truly strange. If 100 watts is the cost of a human life, what is the cost of a single cell? To understand the minimum energy of life, we must look at the construction costs of its smallest unit. Building a cell from scratch requires synthesizing its components, DNA, proteins, lipids, and RNA, from simpler raw materials. Recent comprehensive studies have quantified the Gibbs free energy required to synthesize the biomass of various organisms. The results are staggering in their precision. The most expensive component of a cell, gram per gram, is often its membrane. Lipids are highly reduced molecules, meaning they are rich in hydrogen-carbon bonds. Synthesizing them requires a significant investment of metabolic currency in the form of ATP. Then we have proteins, the workhorses of the cell. They are slightly cheaper to make per gram than lipids, but because they constitute a huge fraction of a cell's dry mass, they represent a massive portion of the total energy budget. The cost isn't just in forming the peptide bonds between amino acids. It is in the synthesis of the amino acids themselves. Surprisingly, the genetic material is relatively cheap to manufacture compared to the membrane and protein machinery. However, the cost of maintaining the accuracy of this information is high. For a simple bacterium like E. coli, the energetic cost to synthesize a new cell is approximately 10 to the power of -11 to 10 to the power of -12 joules. While this sounds minuscule, considering the vast number of bacteria on Earth, the total energy flow through the microbial biosphere is immense. But this construction cost is only paid when a cell divides. What about a cell that is just sitting there, trying not to die? This leads us to one of the most profound realizations in modern biophysics. Life is not just about matter, it is about information. And information processing has a thermodynamic cost. In 1867, James Clerk Maxwell proposed a thought experiment involving a demon that could sort fast molecules from slow ones, seemingly decreasing entropy without doing work. We now know that such a demon must pay an energy cost to measure the speed of the molecules and erase its memory to reset for the next measurement. This connection between information and energy is codified in the Landauer limit, which states that erasing one bit of information costs a minimum amount of energy. Biological cells are full of demons. Molecular machines like pumps and motors often operate as information ratchets, rectifying thermal noise into directed motion. They don't violate the second law because they consume ATP to reset their states. In processes like DNA replication and protein synthesis, errors are fatal. Life needs to distinguish between correct and incorrect molecular partners with incredibly high fidelity. Standard equilibrium thermodynamics limits how good this discrimination can be based on the binding energy difference between the correct and incorrect molecules. However, biology achieves error rates far lower than this limit allows. It does so using a mechanism called kinetic proofreading. Kinetic proofreading introduces an irreversible energy-consuming step into the recognition process. It essentially checks the result twice. If the molecule falls off during the delay, it is rejected. This mechanism dramatically lowers the error rate, but introduces a hidden energy tax. You are paying ATP not to build something, but to ensure you didn't build the wrong thing. This is a purely informational cost of living. Recent theoretical work has identified another invisible cost: the energy required to restrict metabolic flux. A cell is a complex network of thousands of possible chemical reactions. Life requires that only specific pathways are active, while others are suppressed. Thermodynamics dictates that keeping a gate closed against a chemical gradient, or preventing a spontaneous reaction from occurring, requires a continuous expenditure of energy. This restriction cost implies that even a dormant cell is paying a tax just to maintain its metabolic identity and prevent its chemistry from drifting into chaos. If life has to pay these high costs, how efficiently does it spend its currency? The primary currency of the cell is adenosine triphosphate, or ATP. The synthesis of ATP is one of the most studied processes in bioenergetics. The enzyme that makes ATP, F1-pho-ATP synthase, is a rotary motor. It spins at thousands of RPM, driven by a proton gradient. Biophysicists have measured the efficiency of this motor by attaching magnetic beads to it and measuring the torque. The results are astonishing. The mechanical efficiency of the F1 motor is nearly 100%. It converts the electrochemical energy of the proton gradient into the mechanical energy of rotation, and subsequently chemical bond energy, with almost zero waste. It operates at the very edge of what is thermodynamically possible, far outstripping the efficiency of any man-made internal combustion engine or electric motor. However, the overall efficiency of the entire metabolic chain, from glucose to ATP, is lower, estimated at around 40 to 41%. The lost 60% is not entirely wasted. It is released as heat, which for endotherms like us is essential for maintaining body temperature. For single-celled organisms, this heat is largely waste, a necessary tribute to the second law to ensure the reactions proceed at a useful speed. We have looked at active, growing cells. But to find the true minimum energy of life, we must leave the sunlit surface and descend into the Earth's crust. Deep beneath the ocean floor, buried in sediments that haven't seen the sun for millions of years, lies the deep biosphere. This realm is inhabited by microbes that defy our understanding of time and metabolic activity. These organisms, dubbed ionophiles, or lovers of deep time, are not dead, but they are barely alive. In 2020, researchers quantified the power consumption of these deep-sea sediment microbes. The numbers were shocking. Sulfate-reducing bacteria in these sediments survive on approximately 10 to the power of -19 watts per cell. Methanogens, or methane-producing microbes, can go even lower, down to 10 to the power of -20 watts per cell. To put this in perspective, remember that a human uses about 100 watts. A typical surface bacterium uses about 1 watt per gram of biomass. These deep biosphere microbes use 50 billion times less energy than a human. If a human is a bright ceiling fan, a deep-sea microbe is a single photon emitted once every few hours. Their metabolic rates are so slow that they are not growing or dividing in any conventional sense. They are not building new biomass. Instead, their meager energy budget is devoted entirely to maintenance, repairing broken DNA, refolding damaged proteins, and pumping ions to keep the membrane charged. For a standard E. coli in the lab, a generation or doubling time is 20 minutes. For these deep Earth zombies, the turnover time, the time to replace all the carbon in their cells, is estimated to be hundreds or even thousands of years. They are essentially immortal, living in a state of suspended animation, waiting for a geological shift to bring them a fresh meal. This discovery challenges the power limit of life. It suggests that if you remove the cost of growth and reproduction, the cost of mere existence or maintenance is vanishingly small. If deep-sea bacteria are the minimalists of the cellular world, what about viruses? Are they alive in a thermodynamic sense? Viruses have no intrinsic metabolism. They do not consume energy when they are floating in the air or water. In this state, their thermodynamic cost is effectively zero. They are just complex macromolecules, like a crystal of salt. They are thermodynamically stable structures that do not require a power feed to persist. However, they represent a deferred cost. Building a virus requires energy, but that bill is paid by the host. A study on the T4 bacteriophage and the influenza virus showed that the cost to build a T4 phage is roughly equivalent to the cost of a single bacterial cell division. When a virus infects a host, it hijacks the host's ATP. The T4 phage consumes nearly 30% of its host's energy budget during infection, while influenza consumes a mere 1% of a much larger mammalian cell's budget. The concept of zombie viruses has recently moved from fiction to science fact. Researchers have revived Pithovirus and Pandoravirus from 48,500-year-old Siberian permafrost. These viruses had lain dormant, frozen in time for millennia. Unlike the deep-sea bacteria, which were technically on, consuming zeptowatts, these viruses were truly off. This distinction is crucial. The deep-sea bacteria are a non-equilibrium system maintaining a steady state. The frozen viruses are in a trapped equilibrium or kinetically arrested state. Life, as a dynamic process, requires the former. It requires a flow of energy, however infinitesimally small. The power limit calculated for deep-sea microbes of 10 to the minus 20 watts is suspiciously close to the thermal noise floor. If a cell's metabolism is too slow, the random thermal jiggling of molecules will destroy internal structures faster than the cell can repair them. This implies a quantum of life, a minimum flux of energy required to race against thermal decay. This limit depends on temperature. In the cold depths of the ocean or the permafrost, thermal damage is slower, allowing life to survive on less power. This has massive implications for the search for life on Mars, Europa, or Enceladus. We should necessarily look for high-energy biosignatures, like vigorous movement or rapid atmospheric changes. Life on these energy-poor worlds might be cryptobiotic or ionophilic, living on geological timescales, consuming zeptowatts of power, indistinguishable from the background rocks to a casual observer. In the lab, scientists are trying to build minimal cells with the smallest possible genome. Understanding the thermodynamic minimum allows engineers to design synthetic organisms that are ultra-efficient, perhaps for long-duration biosensing missions or biobatteries. To truly appreciate the cost of life, we must perform an energy audit of a living cell. The cell membrane is the defining boundary of life, separating self from environment. Chemically, it is composed of phospholipids. Synthesizing these hydrocarbon chains is chemically reduction-intensive. You are taking oxidized carbon and stripping away the oxygens, replacing them with high-energy hydrogen bonds. Studies using group contribution methods to estimate Gibbs free energy of formation show that lipids have the highest specific energy cost per gram. Membranes are not static walls, they leak. Protons slip through, lipids oxidize. The cell must constantly run bilayer patch operations. In the deep biosphere, where energy is scarce, microbes modify their lipids to be less permeable, effectively insulating their houses to lower the heating bill. Then consider the proteome. Proteins are strings of amino acids folded into precise 3D shapes, making the peptide bond costs 4 ATPs per bond. But synthesizing the amino acids themselves is metabolically expensive, requiring dozens of enzymatic steps. Getting the protein to fold correctly is not always spontaneous. Chaperone proteins often have to actively massage misfolded proteins into shape, consuming ATP in the process. This is a quality control cost. The genome, while cheap to polymerize, has a massive hidden cost in error correction. The error rate in DNA replication is about 1 in a billion. This incredible accuracy is bought with energy via kinetic proofreading. If the cell accepted a higher error rate, say 1 in 10,000, it could replicate much cheaper and faster. But error catastrophe would quickly ensue. The proteins coded by the DNA would be gibberish, and the cell would die. Thus, the thermodynamic cost of the genome is fundamentally linked to the survival of the species. Rolf Landauer showed that information is physical. Cells compute all the time. A receptor binding a ligand is a measurement. Phosphorylating a protein is a memory storage event. Dephosphorylating it is erasure. Transcription consumes about 2 ATP per nucleotide. The Landauer limit is orders of magnitude lower. Biology is seemingly inefficient compared to the theoretical limit, operating at hundreds of times the limit. Why the inefficiency? Speed. The Landauer limit applies to infinitely slow, reversible processes. Life needs to happen now. To compute at a finite speed, you must dissipate extra heat. This leads to the power-speed accuracy trade-off. Active transport pumps act like Maxwell's demons. They sort ions, creating a low-entropy state. The pump changes conformation, driven by ATP. This conformational change ensures the door only opens for the right ion at the right time. The ATP hydrolysis provides the energy to reset the demon for the next cycle. Molecular motors like kinesin use thermal noise to move. They don't fight the storm of water molecules. They use the storm, rectifying random jiggles into forward steps by burning ATP to prevent backward steps. They are judo masters of thermodynamics. The discovery of the deep biosphere changed our definition of metabolic limits. Below the ocean floor, there is no light. Oxygen is rare. The only food is fossil organic matter that settled there millions of years ago. It is a starving ecosystem. These microbes do not compete, they endure. They have stripped down their machinery to the absolute basics. They express only the genes needed for repair. Over time, the amino acids in proteins spontaneously flip from left-handed to right-handed. This is a purely entropic decay process called racemization. Deep biosphere microbes spend a significant chunk of their tiny energy budget just swapping these amino acids back. It is a Sisyphean task of molecular housekeeping. Tardigrades, or water bears, can enter a ton state, desiccated and dormant. In this state, metabolism drops to near zero. They replace the water in their cells with specific proteins and sugars that turn into a biological glass. This arrests molecular motion. Without water, chemistry stops. Thermal decay slows to a crawl. This suspended animation proves that life is not a continuous fire. It can be paused. The thermodynamic cost of being a structure is different from the cost of running a structure. The ton state pays almost no running cost, relying on the structural stability of the glass to resist entropy. Evolution has also constrained human limits. Evolutionary anthropologists suggest that humans evolved a constrained energy expenditure model. If you exercise more, you don't necessarily burn more calories in the long run. The body adapts by shutting down other systems, like immunity or reproduction, to keep the total budget constant. This is why the 2.5 times BMR limit exists. Evolution designed us to handle spikes in activity, but not chronic high output. We are optimized for efficiency, not maximum throughput. The human brain is 2% of body weight but consumes 20% of the energy. Why so expensive? Ion gradients. Neurons must constantly pump sodium and potassium to be ready to fire an action potential. It is the cost of readiness. Firing a spike is expensive, but resetting the neuron after the spike is where the bulk of the energy goes. This is the biological equivalent of the erase step in Landauer's principle. Prigogine described life as a dissipative structure. Life exists only because it is far from equilibrium. The further you are from equilibrium, the more energy you must dissipate to stay there. Some theories suggest systems settle into a state where they produce the least entropy possible, given the constraints. The deep biosphere microbes seem to exemplify this. They hug the equilibrium line as closely as possible without crossing it into death. Ultimately, the story of life's energy is the story of the electron. From the high-voltage drop of sunlight in a leaf to the low-voltage trickle of sulfate reduction in the deep earth, life is an electron transport phenomenon. We are, in the words of one physicist, a way for the electron to find its ground state a little faster. But in doing so, we build cathedrals of complexity. The minimum energy of living matter, that 10 to the minus 20 watt flickering in the dark, is a testament to the tenacity of this complexity. It proves that life does not need a roar of fire, it just needs a spark. As we look to the stars and into the microscopic depths, we now know what to look for. Not just the heat of the living, but the faint, stubborn warmth of the barely alive.